Our lab is primarily focused on the molecular biological effects of ultrasound, and we have a particular interest in using them for regenerative medicine and stem cell therapies. Now, we use mostly mesenchymal stem cells. They downregulate inflammation, stimulate endogenous repair mechanisms, and it's been previously shown that MSC infusions can improve acute kidney injury. And what we did was publish a paper last year that demonstrated if you pre-treat kidneys with pulse-focused ultrasound before injecting the cells, that we saw better AKI outcomes, but we were unclear on what the mechanism is. So this is just a quick background slide. As we've been hearing quite a bit in this meeting, the biological effects of depositing ultrasound into tissues really is extremely complex and poorly understood. And what we show here is this is the molecular response in the kidney after giving pulse-focused ultrasound. And what we show is that if you inject mesenchymal stem cells intravenously in the middle of this, that they can respond to these molecular cues and they will actively extravasate into the parenchyma. And this is a molecularly driven event. And what we published last year was that in an acute kidney injury model, pulse-focused ultrasound causes greater homing of MSCs. And the correlate with that is an improvement in renal function and survival by coupling pFUS with MSC treatment compared to just MSC treatment alone. One thing that we noticed was that the stem cells homing to the pFUS-treated kidneys produced a disproportionately high amount of interleukin-10 that couldn't be explained simply by enhanced homing. So we did a proteomic study on the AKI kidney, and we found that pulse-focused ultrasound upregulates interferon gamma. And this immediately drew a connection that Joe Frank talked about a little earlier in the cardiovascular section, where stem cells can be preconditioned. So MSCs exposed to pro-inflammatory cytokines often become more potent therapeutics. And so we wondered if there was this interferon gamma IL-10 axis, and we were actually controlling that with pulse-focused ultrasound. So the first experiment I want to show you here is just that this axis does exist in AKI, that we can culture MSCs and add recombinant interferon gamma, and this causes those MSCs to produce more IL-10. If we inject these two types of cells into mice that have AKI, we see homing to the kidneys in about similar numbers, but there is greater amounts of IL-10 in the kidneys that receive the stimulated MSCs, and corresponding improvements in renal function. So to examine the role of pulse-focused ultrasound in this, the first thing we did was to use an interferon gamma knockout mouse. And what we see here is that there's really no interferon gamma in the mouse. Our assay was working. This is the wild type and shows that pulse-focused ultrasound does increase it. But if you inject cells following pulse-focused ultrasound into the interferon gamma knockout mouse, you get more cells homing, but in the absence of the interferon gamma, you don't see more IL-10 made by these cells. And in turn, the combination therapy fails to yield the additional improvements in renal function. The other experiment is we can turn things on its head and go back to the wild type mouse, but this time use IL-10 deficient cells. So these were given siRNA to knock down IL-10. And what we see again is that greater homing to the pFUS treated kidney. They don't make IL-10, so we don't see any increase in that. And lastly, with renal function, the additional gains with the coupled therapy do not present themselves, and the combination with the knockout cells is the same as knockout cells alone or wild type cells alone. So with that, we can conclude that pulse-focused ultrasound does increase renal interferon gamma, which does stimulate the MSCs which home to those kidneys to further improve acute kidney injury. And this by definition makes pFUS a neoadjuvant, and this has major implications in cell therapy, where this really is about the first demonstration where we can control cell potency through non-chemical, non-genetic means in vivo. So with that, I'll go ahead and take any questions anybody has. Thank you. Oh, I'm sorry, one last thing. We are hiring for postdocs, so if you are interested in the kind of work you've seen from our group, please talk to myself or Joe Frank. So thank you. With that, I'll take any questions.